This is Sally Schessler, Director of Education for Allergy and Asthma Network, and it's my pleasure to welcome you to this month's webinar in our 2019 webinar series titled Advances in Allergy and Asthma, brought to you in partnership with the American College of Allergy, Asthma, and Immunology. Our webinar series helps Allergy and Asthma Network live out our mission, and that mission is to end the needless death and suffering due to asthma, allergies, and related conditions through outreach, education, advocacy, and research. Dr. Maeve O'Connor joins us for today's webinar, and she will be speaking about microbiomes and their role in allergy and immunology. Dr. O'Connor provides a more progressive approach for treating allergy, asthma, and immunology patients. She's board certified by both the American Board of Internal Medicine and the American Board of Allergy and Immunology. She is one of only 3 specialists to have completed the University of Arizona Fellowship of Integrative Medicine and has been selected by her peers to be included in the Best Doctors in America database from 2011 to today. Dr. O'Connor has been one of Charlotte's Magazine's top doctors for both adult allergy and immunology and pediatric allergy and immunology from 2007 to the current. Additionally, Dr. O'Connor is listed in North Carolina's Best Doctors. She treats patients of all ages and is fluent in Spanish. Dr. O'Connor was the first board-certified allergist in the region to offer sublingual immunotherapy. She is the principal investigator on numerous clinical trials and has published research articles on desensitization, immunotherapy, asthma, and food allergy. She is currently involved in the clinical research of asthma, allergic rhinitis, immune deficiency, nasal polyposis, atopic dermatitis, immunotherapy, and novel treatments for atopic diseases. Dr. O'Connor is a fellow of the American College of Allergy, Asthma, and Immunology and lectures extensively on all subjects involving allergy immunology locally, nationally, and internationally. Dr. O'Connor, thank you so much for being here with us today and for sharing your expertise.
Thank you so much, Sally. It's a pleasure to be here and an honor to be invited. I'm very excited about this topic. Today we're going to be talking about the... I can't search the web on Apple Watch. Today we're going to be talking about an overview of the microbiome and its impact on inflammatory disease. This is the first portion of our presentation today. Now, what is the human microbiome? We all are starting to hear a lot more about it. It's become a very hot topic. And I just want to give you a little bit of information on this, this definition. The microbiome is defined as the sum of these microbes that are shown here in this slide. They're genomic elements and interactions in a given ecological niche. The composition and diversity of the microbiome varies across body sites, as you can see here. And this results in a series of unique habitats within and between individuals that can change substantially over time. The establishment of stable microbial communities closely tracks host growth and immune development during the first few years of life. Factors that influence this evolution include many things, including antibiotic use, birth mode, infant nutrition, as well as infant biodiversity in the home, surrounding environment, and family member exposure. Delayed or altered establishment of these microbial communities leads to microbiome immaturity and has been associated with increased risk of allergies and asthma later in life. By young adulthood, both humans and other mammals support one of the most complex microbial ecosystems on the planet, with over 100 trillion bacteria comprised of potentially many thousands of microbial species in the distal gut. That's pretty impressive to me. Now, that is what we are talking about today, the human microbiome. Our learning objectives are listed here. We're going to describe the typical composition of the human microbiome and the functions that it supplies to the host. We're going to summarize evidence that the human microbiome plays a role in inflammatory and allergic diseases. We're going to identify mechanisms by which bacteria modulate the immune system. And we're also going to name cutting-edge technologies for exploring microbiome composition, function, and the role in disease. That is just an introduction to what we're going to be doing today. I'm just checking the battery on my computer. Give me one moment. Okay. So how do your microbes influence your health? This is kind of a quiz question. Obviously, you are all on mute and won't be able to answer the question, but we're going to go through the answers. So, do microbes influence your health by competitively excluding pathogens, by metabolically transforming your diet and your drugs, by modulating the immune system, or all of the above? Giving you a second to think about this. The answer is all of the above. Microbes influence your health by all of these things: competitively excluding pathogens, metabolically transforming your diet and drugs, as well as modulating the immune system. So how do your microbes influence your health? They really do this in two ways. First of all, they do this by, quote, taking up real estate, protecting against pathogens, and also protecting against antibiotic-associated diarrhea and vaginal yeast infections. A second way that microbes influence your health is through their metabolism, by extracting extra calories from your diet, producing health-promoting metabolites, producing detrimental metabolites as well, and metabolizing drugs. Everyone on this call has heard of the hygiene hypothesis and is very familiar with this, I am sure. But this discussion today is going to take this just a little bit further. The third way that microbes influence your health is by influencing your immune system. Influencing your immune status as an adult, as we age and develop inflammatory diseases. Also training your immune system as an infant, allergy, asthma, and autoimmune disease. So the question is, should we let them eat dirt? Is poop exposure okay, as is looking at this picture of this cute little child on this slide? Although in the front of it, it looks like those might be chocolate candies masquerading as poop. But the data is clear that clean environments may set kids up for immune problems long term. And I want to repeat that one more time because I see a lot of parents in the office and they're very concerned about if a piece of candy falls on the floor or if something is on a table. And I want to just repeat it one more time. The data is clear that clean environments may set kids up for immune problems long term. So this slide is looking at the highly sophisticated mucosal immune cellular and molecular networks that need to be constantly coordinated in order to tolerate the presence of a large number and diversity of bacteria, while protective immune responses to potential pathogens must be maintained and induced on demand. The balance between immune tolerance and inflammation within tissues is regulated in part by the crosstalk that occurs between immune cells and the microbiome. Any disruption in the communication between the microbiome and the host due to altered microbiome composition and or metabolism is thought to negatively influence immune homeostatic networks. And you can see here on this slide is depicting that balance, that homeostasis that is necessary for a healthy life. By virtue of its responsibility to recognize, respond, as well as adapt to countless foreign and self molecules, the immune system is central to the processes of health and disease. While the classical view of the immune system is that it evolved to protect us and protect organisms from infection by microbial pathogens, we actually peacefully coexist with our vast and complex microbiota. In fact, as shown here, the magnitude of the molecular interactions between the microbiota and the host immune system appear to be extensive and a constant, delicate balancing act as demonstrated here. This is actually a really good slide because it shows how we coexist with our microbiota and how our immune system and that delicate balance that must be maintained for appropriate health. The gut microbiota seems complicated, but it has simple composition at the phylum level. The gut microbiota helps us combat aggressions from other microorganisms, maintaining the wholeness of the intestinal mucosa. That intestinal mucosa is so important. It plays an important role in the immune system, performing a barrier effect. A healthy and balanced gut microbiota is key to ensuring proper digestive functioning. And at the phylum level, as shown here, the gut microbiota composition is very simple. However, it becomes more complex. When you look at a species level, the microbiome becomes extremely complex. The many species within each phylum have different biological properties, and significant changes detected at higher taxonomic levels are likely driven by only a subset of the species in those higher taxa. And if you see here, we illustrate the high diversity and variability among individuals and the degree to which taxonomic grouping at high levels can mask this diversity using 16S rRNA sequencing data. And this was published in Nature in 2012, showing the difference again between the gut microbiota being simple at the phylum level, but complex at the species level. So the large effect of age on the human gut microbiota is shown here, and it's consistent with studies that have detailed dramatic changes in the gut community composition over the first 3 years of life. There are many studies that have shown this, published in 2007, 11, as well as 2012. Studies of the early assembly of the gut microbiota, whether longitudinal or cross-sectional within the United States, or even across countries representing distinct cultural traditions, indicate that the infant microbiota undergoes a successful trajectory until a relatively stable adult-like configuration is established. Our results further show here in this slide that the early gut microbiota more closely resembles other body sites, including the vagina and skin, than the fecal microbiota of Western adults. This is consistent with studies of the effects of delivery mode on the microbiota of newborns, which showed that vaginally delivered babies had microbiota resembling the mother's vagina across multiple body sites, and babies born by C-section had an initial microbiota resembling skin only. However, given that the progression of the infant gut microbiota to one resembling an adult occurs slowly over the first 1 to 3 years of life, during which time exposures to gut microbes occur, the trajectory from infant to adult gut microbiota is likely not only due to a replacement of initial colonizers. Another possible contributor to consider is shared biological or physiological characteristics between the vagina and infant gut, such as more similar environmental stressors, which results in the selection of some of the same types of bacteria. But the important point here is that healthy gut microbiota composition does differ with age, and many different things can affect this. What about culture and with diet? Healthy gut microbiota also has been shown to differ with culture and with diet, as depicted here. The separation of the gut microbiota of Western cultures and Italy from the gut microbiota of individuals in the developing world, such as Burkina Faso, Malawi, and Venezuela, supports the conclusion that strong and consistent differences exist between the gut microbiota of individuals in Western cultures compared with agrarian cultures. The clustering of fecal samples from individuals from Burkina Faso, Malawi, and Venezuela is consistent with independent reports of enrichment of Prevotella in fecal samples from individuals living in non-Western societies compared with Italy or the United States. The finding that Prevotella could predict community-wide diversity patterns is not surprising, given the observation that a trade-off between Prevotella and Bacteroides are associated with overall stratification of diversity, both within Western populations and in cross-cultural comparisons. And these bacteria names are complicated and hard to pronounce, but I hope that you're picking up what I'm trying to say about the differences in the bacteria and how this affects the overall gut microbiota and the patient or the person in general. So we now understand that early life microbial exposures definitely influence allergy, asthma, and immunity later in life. There are known associations, as we're all familiar with, between early life disturbances and antibiotic use, C-section delivery, specific farming environments, household pets, whether they are furred or non-furred, specific microbial dust that can be found in mattresses and pillows, and we're going to get into that in a little bit later, as well as the overall gut microbiome. And on the right of this slide, you can see some germ-free or notobiotic mice, which have been either stripped of all their microbiota or every bacteria has been identified, which we don't have the luxury of doing necessarily in humans. So for this portion of the presentation, the conclusions are the human gut microbiome is complex. I certainly have learned a lot in preparing this presentation. Lots to learn, but we have learned a lot. The gut microbiome influences health by a variety of mechanisms. It can have profound effects on health, in fact. And microbe-targeted therapies are a promising research line that is still in its infancy. At the beginning of the presentation, just before we went live, I was talking about how I hope that there are some medical students, fellows, residents out there listening to this, because there are so many areas with the microbiome that we need to target so we can learn more and take better care of our patients. So now we're going to look at the intersection of the microbiome, diet, and inflammatory disease, and of course, my specialty, allergy, asthma, and atopic eczema. Learning objectives for this portion. We're going to summarize how diet influences inflammation in different disease contexts, including airway disease. We're going to describe how microbes impact the effects of diet directly through the metabolic transformation of dietary components, and how microbes impact the effects of diet indirectly through their effects on signaling pathways. And we're going to summarize research on dietary intervention as a therapeutic strategy. It all goes back to Hippocrates, who said that food was the medicine and medicine was the food. So we're going to talk a little bit about that today. We're going to take a deeper dive into the interaction between diet and the microbiome, as I mentioned. We know that the diet impacts the microbiome composition and that the microbiome metabolically transforms the diet and consequently the host phenotypes. There are many diseases like heart disease, allergy, asthma, diabetes, whether it's insulin resistance, glucose intolerance, even Clostridium difficile infection. All of these can be impacted by the diet. As a matter of fact, diet is one of the most influential factors on the gut microbiome composition differences. This slide here, for example, that was published in 2018 by McDonald and colleagues, shows the American Gut Project, which analyzed samples of over 10,000 people. So a big number of people were studied. The self-reported dietary data suggested unexpectedly that the number of unique plant species that a subject consumes is associated with microbial diversity, rather than self-reported categories such as vegan or omnivore. And you can see the difference here by looking at figure C as well as figure E. But figure E also shows that plant consumption was also associated with a reduction in certain antibiotic resistance genes. Individuals who consume more than 30 types of plants per week, compared to those who consume 10 or fewer plants per week, had significantly reduced abundance of antibiotic resistant genes. Everyone on the call probably remembers that in the olden days, antibiotics were given out all the time, left and right, until we started realizing about antibiotic resistance. But this study showed that the patients who ate the most plants per week had reduced abundance of antibiotic resistance genes. Now, just shifting gears a little bit, this shows that the short-term consumption of diets composed entirely of animal or plant products does alter microbial community structure, and overwhelms inter-individual differences in microbial gene expression. The animal-based diet increased the abundance of bile-tolerant microorganisms such as Allistopus, Bilophila, and Bacteroides. The animal-based diet also decreased the levels of firmicutes that metabolize dietary plant polysaccharides such as Roseburia, Eucobacterium rectale, and Ruminococcus bromley. Microbial activity mirrored differences between herbivorous and carnivorous mammals, reflecting some trade-offs between carbohydrate and protein fermentation. So foodborne microbes from both diets transiently colonize the gut, including bacteria, fungi, and even viruses. And finally, increases in the abundance and activity of Bilophila wadsworthia on the animal-based diet support a link between dietary fat, bile acids, and the outgrowth of microorganisms capable of triggering inflammatory bowel disease. In concert, these results demonstrate that the gut microbiome can rapidly respond to altered diet, potentially facilitating the diversity of human dietary lifestyles. So maybe we are what we eat after all. This next study by Trompet and colleagues found that dietary fermentable fiber content changed the composition of the gut and lung microbiota. In particular, it altered the ratio of firmicutes to Bacteroides. Those are two different bacterial names. The gut microbiota metabolized the fiber, which then led to the increased concentration of circulating short-chain fatty acids, or SCFAs. Mice fed a high fiber diet had increased circulating levels of SCFAs and were protected against allergic inflammation of the lung, whereas a low fiber diet decreased levels of SCFAs and increased allergic airway disease. And this slide here demonstrates how the diet can impact heart disease as well as kidney disease and other diseases, resulting in inflammation that leads to atherosclerosis, coronary thrombosis, adverse cardiac remodeling, and chronic kidney disease. I think this slide, this study from 2018, and particularly this slide, is very important for us to understand because, again, what we're putting in our bodies is what can be leading to alterations in the microbiota, and that can lead to these chronic diseases that are causing morbidity and mortality in the United States and across the world. And the conclusions here, as I said, is diet shapes the microbiome. Microbial metabolic activities do transform the diet. Microbes, whether they're doing it directly or through their metabolic activity, drive health and disease effects of diet in many disease contexts, whether it's metabolic disease, whether it's allergy, asthma, or immune dysregulation, cardiovascular and renal disease, as we saw. But what a healthy gut microbiome is may be context specific and does depend on the diet, and may also be individually specific, which we're going to get to a little bit of that in a moment. Okay, shifting gears again. I'm going to talk about the microbiome and atopic dermatitis, another disease that's near and dear to my heart. We're going to look at the innate immune control of the microbial pathogenesis, as well as the role that Staph aureus plays in atopic dermatitis. Finally, we're going to explore how the microbiome interacts with Staph aureus in atopic dermatitis. So, the skin optimizes microbial interactions. If you look to the left side of this slide, if the skin surface looked like this, where there were no disruptions or holes, if you would, in the skin surface, then the standard surface area of the skin would be an estimation of 2 meters squared. But if you look to the right of the slide, you see what a typical human skin typically does look like. We have appendage openings, we have breaks in the skin, cracks in the skin, and patients with atopic dermatitis even have more of these than somebody who does not suffer from atopic dermatitis. Therefore, the surface area is closer to 25 meters squared. The bottom line is we have billions of opportunities for the skin microbiome to interact with the outside world as well as the immune system, whether we do or do not have atopic dermatitis, but you can see the huge difference for opportunities for immunological assaults for patients who do have atopic dermatitis. Microbial fitness does not necessarily depend on host health. The host must innately control microbial growth. We call it microbe fitness, quote unquote. It's constantly fluctuating between health and disease, as demonstrating here. And this is something that was published in 2013 by Sanford and Gallo, a beautiful representation of the delicate balance that must occur between the microbes, the host, and again, that microbial fitness. And this image here depicts how the immune system is trying to always exert pressure for beneficial interactions. We want mutualism, we want commensalism, we want benefit, and our immune systems are constantly fighting to have benefit. Unfortunately, we do have things on the outside of the world that are trying to be to the immune system's detriment, such as predation, parasitism, commensalism, and competition. And when those things occur, the immune system fights to get to the left side of the slide to be beneficial, but sometimes is unable to do that depending on the strength of that immune system. But we see that constant struggle to develop, going back to this slide for one moment, that microbial fitness, and is not always dependent on just host health. And the host must innately control microbial growth. Moving on to another subject, the key components of innate immunity: human host defense, antimicrobial peptides and proteins, otherwise known as AMPs. They play a very critical role in warding off invading microbial pathogens. In addition, AMPs can possess other biological functions, such as apoptosis, or what we also refer to as programmed cell death, wound healing, and immune modulation. This provides, over here you can see in this slide, an overview on the identification, activity, the three-dimensional structure and mechanism of action of human AMPs selected from the antimicrobial peptide database. Over 100 such peptides have been identified, including LL37, HBD2 and 3, and many, many more. And they've been identified from a variety of tissues and epithelial surfaces, including the skin, eyes, ears, mouth, gut, immune, nervous, and urinary symptoms. So again, it's a very complex system that keeps us in homeostasis. AMPs are essential for the defense of the skin. And this shows on the left of the slide, you see normal skin, you see a skin that has been infected with strep. The brown staining is looking at those AMPs that I have just described to you. And then 12 hours later, the wound healing. And you can still see the prevalence of brown staining, showing that the essential role of AMPs in the defense mechanism of the skin. The AMPs are doing their job there to get the skin back to homeostasis. And you can see on the right side of the slide, mice who do have AMPs that are working and mice that have AMPs that are not working. Wound healing versus poor wound healing. So this is summarizing what we were just talking about, looking at AMPs and the host control of microbial growth. And you can see that we are just scratching the surface when we're talking about AMPs. This is a very complex slide looking at the epidermis, dermis, as well as the adipose tissue. And you can see all of the AMPs that are involved, again, in maintaining homeostasis in normal skin, but how dysregulation of this can cause problems in a patient who has atopic dermatitis. And this slide here highlights the role of AMPs to two different ways that these AMPs help have host control of the microbial growth. On the left side of the slide, you can see that there's direct killing of the microbes with electrostatic interaction, membrane rupture, inhibition of intracellular function, which leads to bacterial lysis. On the right side of the slide, you can see the other way that AMPs help host control of microbial growth with modulation of host immunity, whether it's through chemotaxis or toll-like receptor response. With chemotaxis, we're looking at the neutrophils, the T cells, the monocytes, the dendritic cells. They may recruit or activate immunocytes to help modulate that host immunity. And when we're looking at the TLR response, we're looking at other mechanisms and crosstalk that can neutralize bacterial products to suppress inflammation and or enhance nucleic acid recognition to promote autoinflammation. So this is how AMPs work to keep us healthy. This is a New England Journal of Medicine article that shows that the deficiency in the expression of AMPs, again, antimicrobial peptides, may account for the susceptibility of patients with atopic dermatitis to have and continue to have infections with Staph aureus. And this was a hallmark article that was published in 2002. And as you can see from this graph here, you can see that Th2, or the more, quote, allergic cytokines, suppress AMPs, or AMP expression. And this slide here shows that multiple studies over the last 45 years have reproducibly showed that Staph aureus is clearly associated with atopic dermatitis in both culture-based and DNA-based analysis of patients' skin with atopic dermatitis. And this was just from 1974 to 2012, this has reproducibly been shown. And then moreover, these studies by Nakatushi demonstrated Staph aureus is present in patients with atopic dermatitis, and this was published in 2016. You can see, if you look closely, the non-atopic dermatitis skin versus the atopic dermatitis non-lesional versus lesional skin, and you can see that Staph aureus in human dermis is definitely associated with active atopic dermatitis. Now, interestingly, recent studies also suggest that the presence of Staph aureus precedes the development of atopic dermatitis even as early as 2 months of age. And these are two studies that were both published in 2017 that show this. One entitled The Skin Microbiome Before Development of Atopic Dermatitis, Early Colonization with Commensal Staphylococciat at 2 months is associated with a lower risk of atopic dermatitis at 1 year. And then Staph aureus precedes development of AD skin or atopic dermatitis skin colonization by Staph aureus preceding the clinical diagnosis of atopic dermatitis in infancy. So the staph has been shown to be there prior to the development of active atopic dermatitis lesions. And there are some plausible molecular explanations for this. As you can see from this slide, this is a complicated slide, but the important thing is to just look at the epidermis versus the dermis. And you can see, again, the communication, the relationship between the immune system and the presence of Staph aureus in these patients with atopic dermatitis. The key is there is likely a very important causal association between staph and atopic dermatitis. And as you can see in the dermis of these patients with atopic dermatitis, there's an increased IL4 as well as IL13, increase in IL17, and increased IgE in the dermis. And as we were looking for ways to target therapies for patients with atopic dermatitis, this slide is very important because we try to find ways to make these patients have healthy skin. But this again is a plausible molecular explanation for that causal association between Staph aureus and atopic dermatitis. So does blocking IL4 improve the skin microbiome? If you look at the data that is shown on the slide here, again, a complicated slide, which I'm going to try to simplify for you. It is basically, it appears that blockade of IL4 does improve the skin microbiome. As you can see here, when you look at the placebo group versus the treated group, and when you look at lesional skin versus non-lesional skin. Clearly, blocking IL-4 decreases Staph aureus colonization as shown here, and this occurred as soon as week 4 and continued beyond week 16. And this is looking at PCR for Staph aureus. So they weren't letting the Staph aureus hide at all. And again, this was as early as week 4 and continuing beyond week 16. So blocking IL-4 definitely decreases the staph colonization and may improve the results for patients suffering from atopic dermatitis. We're talking about homeostasis and we're talking about balance earlier in this presentation, and here I just want to talk about the ongoing cycle of Staph aureus in atopic dermatitis. The microbiome resulting in inflammation, proteolysis, Th2 or allergic skewing, which results in skin barrier defects and decreased AMPs, our friend the AMPs, which in then return changes the microbiome. And you can see here this kind of ongoing cycle of the Staph aureus and the atopic dermatitis and the filaggrin gene and all the things that can skew the skin so that it does not heal well in atopic dermatitis. Okay, we'll take a 5-second stretch because this is a complicated but exciting topic. Can microbes protect from asthma? We're going to look at very important studies comparing Amish and Hutterite gut microbiota in the next following slides. So again, the New England Journal of Medicine. This is a study that looked at the innate immunity in Amish and Hutterite farm children and found that despite the very similar genetic ancestries and lifestyles of Amish and Hutterite children, the prevalence of asthma and allergic sensitization was 4 and 6 times as low in the Amish, whereas median endotoxin levels in Amish house dust was 6.8 times as high. So differences in microbial composition were also observed in dust samples from Amish and Hutterite homes. Profound differences in the proportions, phenotypes, and functions of innate immune cells were also found between the two groups of children that were thought to be very similar, again, as far as their genetics, their ancestry, and lifestyles. So what is the farm effect? The farm effect involves the environmental microbiome. In the ALEX study, endotoxin levels in child's mattress dust were inversely related to the occurrence of hay fever, atopic asthma, as well as general atopic sensitization. Blood cells from ALEX farmers' children expressed significantly higher levels of the microbial sensors CD14 and TLR2 than those from non-farmers' children. In the Parasophil and Gabriella studies, children who lived on farms had lower prevalence of asthma, and were exposed to a greater variety of environmental microorganisms than children in the reference group. And interestingly, diversity of microbial exposure was inversely related to asthma risk. And there are pretty significant statistical odds ratios, et cetera, that are listed there for you on the slide. Farm exposure was positively associated with bacterial diversity or richness. We call it both terms are kind of interchangeable. And this was in the mattress dust samples. And asthma was inversely associated with that microbial richness in mattress dust. So, you know, maybe Dr. House was onto something on that show when he would go to people's homes and measure what was going on in their houses. Maybe we need to start measuring microbial richness in mattress dust of our patients. So is the host microbiome involved in asthma protection? And that's a question that would obviously need to be answered. And, you know, there are strong relationships between composition of the gut microbiome and the risk of childhood asthma. The gut microbiome of infants who were 100 days of age or less who develop asthma has been shown to be depleted in Faecalibacterium, Lachnospira, Veillonella, and Rotheria in a study published in 2015 by Arrieta and colleagues. The neonatal, so less than 35 days of age or less, gut microbiome harbors 3 distinct compositional states, one of which was especially associated with later development of asthma and showed a lower relative abundance of certain bacteria. And these bacteria, for example, were Bifidobacterium, Akkermansia, and Faecaliobacterium, and a distinct fecal metabolomiome enriched for pro-inflammatory metabolites. And this was published in 2016 by Fujimura and his colleagues. Another important point and important study that was published in 2018 was that delayed maturation of the gut microbiome at age 1 year, is associated with risk of asthma development at age 5 years in children who are born to asthmatic mothers. So we know that there's the risk of just being born to an asthmatic mother, but now we have to think about that microbiota, the microbiome. And I'm not going to go into too many details on this slide, but just to show the distinct gut microbiota in the Amish and Hutterite children, they are very different, although again, these populations of children thought to be very similar on upbringing and exposures. But you can see here, when you look, they are actually quite distinct. So the results of these studies show that the host microbiome is definitely involved with asthma protection. And one important point is when transferred to germ-free mice, Amish, but not Hutterite gut microbiota, inhibit allergen-dependent allergic hyporesponsiveness, ovospecific IgE and IL-13 expression, and preferentially induce lung-resident eosinophils. Secondly, specific modules or consortia of Amish gut microbes are associated with asthma protection phenotypes and are endowed with distinct, relevant functional capacities. Importantly, this just shows that the Amish environment seems to provide protection against asthma by engaging and shaping the innate immune response. So I've hit you with a lot of, I hope, interesting and exciting information, and I tried to keep it at 45 minutes. I would be happy to engage any questions. There's a lot of information that I've presented, and there's a lot of information that we need to still research. I appreciate your attention, and thank you so much for allowing me to present this exciting information to you.
Thank you so much, Dr. O'Connor. We do have some questions now for you. The first one is, can we do anything preventative if we need to take an antibiotic, or how can I improve my microbiome after antibiotics?
Well, as you mentioned, Sally, when you introduced me, and thank you for that nice introduction, I am also integrative medicine trained. And so one thing that we think about when we give somebody an antibiotic is that gut health, right? We want to have the gut be healthy. So we want to make sure that if we do prescribe an antibiotic, that somebody is on a probiotic, that they are eating a healthy diet, and that they are using just normal universal precautions. Washing your hands with soap and water. Don't need to have a bunch of hand sanitizer. If something drops on the floor, you can still eat it. But with antibiotics, you definitely want to make sure that you protect the gut by being on probiotics and having a healthy diet.
Okay, well, so now the question relates to probiotics. For the therapies for microbiome, does that refer to the administration of probiotics to patients?
I'm sorry, repeat that again? I just missed the first part.
They were asking if with microbiome therapies, is that revolve around giving probiotics to patients?
It depends on the patient, but I definitely think that probiotics are good for most people, because most people aren't following the diet that I outlined when I was reviewing those studies. And most people are not living the healthy, stress-free lifestyle that we're supposed to live to keep our homeostasis or our balance. So I definitely recommend a good probiotic for most patients. Yes, I do.
I didn't know we were supposed to be living a stress-free life. I guess I got to think about that.
It's supposed to be.
Another question is, I have eczema and someone told me to put yogurt on my eczema. Have you ever heard of anyone doing this?
I have never heard of anybody doing that, but it's not a bad idea when you think about the fact that the microbiota could be skewed. You have to worry about Staph aureus. And good healthy bacteria can commensally help us have homeostasis, whether it's not in our gut, whether it's in our whole body, or topically on the skin. I'll have to research that further. Interesting idea.
Isn't it though? Yeah, it's great. Okay, so next question is, can humans improve their gut microbiome eating things such as Greek yogurt, kombucha, I don't know how to say that one, etc., or by taking over-the-counter products claiming to improve the gut microbiome?
So what I will say here is, the answer that lots of people don't love is it's definitely most important to always just start with the basics. Rather than running out and buying a whole bunch of products, start with a healthy diet, healthy lifestyle, an anti-inflammatory diet. We saw the effect of plants on the diet versus an animal fat-based diet. So I would definitely start there. In addition, I would recommend healthy multivitamin, probiotic, and Greek yogurt is a great way to do that because you can get it through food. Kefir is another way. It's even a more dense kind of Greek yogurt. I've had patients who go on that and their inflammation goes down. They itch less, they sneeze less, they wheeze less. But I always want to kind of caution patients, don't run out and just start buying a bunch of stuff. Start with the basics and just live a healthy lifestyle and then add things if you need to. Depending on how busy you are, depending on your lifestyle, your work style, you may have to go out and buy some of those products. And if so, if anyone wants to email me, I do have a list of some products that I recommend.
Wonderful. Someone's asking if you would sum up your presentation in just a few bottom line comments. And I know one of them is you don't need to worry about having too clean of an environment. But if you could just kind of give a few just a few bullet points about your presentation, this person would appreciate that.
Absolutely. So, we live commensally, or we're supposed to live happily with the bacteria that are in our body. And what we do to our bodies, what we eat, our lifestyle, stress, working too much, constant screen time, all of that skews our immune system. And we used to think it was just the immune system, but now we know that it's also due to those bacteria that we're supposed to be living happily with. If we're not living happily with those bacteria, we can get in trouble. Our immune system can get turned on. And when our immune system gets turned on, it can lead to bad things like allergy, asthma, cardiac disease. So to sum it all up, it's better to be dirty than clean. It's better to have a healthy lifestyle and eat clean. And don't forget, your gut is very important to everything else in your body. And when you go back to that original, the very first slide I showed that showed the microbiota of all the different parts of the body, don't forget it's a lot more complicated. But if you keep it simple with your lifestyle, you can be healthy and your patients can be healthy.
Oh, that was great. Thank you. Well, this kind of piggyback right on what you said is, how do you advise mothers on whether or not to let their young children play in the dirt or in less than immaculately clean environments?
Play in the dirt. I will even give a personal story. My child, who is now almost 8 years old, for the first 6 weeks of his life, he was very healthy. He went into daycare. The next 6 weeks, he was pretty unhealthy, got sick a lot. And then after that, he has been one of the healthiest children I've ever known in my life. He's had one infection since then, and he's almost 8. So I feel that exposure, when he was kind of like, sometimes people call daycare a little bit of a petri dish, I think it was the best thing I ever could have done for him, because I think it strengthened his immune system. And I think his microbiome and his immune system are living together just fine.
Oh, yeah, that's wonderful. I have to tell you, in my experience of being a school nurse, I was never healthier than when I was in the school health office with everything coming at me all the time. So
Yeah.
I'm sure it had something to do with exactly what you've been talking about. So
Yeah,
next,
I was just going to say, so, again, I go back to that picture of the little boy that could have been chocolate, could have been poop in that picture.
Let's pretend it's chocolate, okay?
Either way, though, you know what? It's good to have exposure to the things that keep us in homeostasis. We need that balance, Sally. We got to have that balance.
Okay, but I still want to think about it as chocolate. It's too close to the dinner hour, Okay, next question is there any association of altered microbiome due to GI illness or antibiotic use and increased asthma exacerbations?
I don't know of any studies off the top of my head, but I think if you think about overuse of antibiotics, we know that those lead to multi-drug resistant bacterial strains. And we know that if you're on long-term antibiotics, it can lead to that C. diff, that Clostridium difficile that I showed on one of the slides. So I definitely believe that overuse or misuse or incorrect use of antibiotics can affect the microbiome, and that can definitely affect overall health. But I don't know of any specific studies off the top of my head right now.
Okay, someone's asking, can any of the new biologic medications help with keeping our microbiomes healthier?
Well, there have been no studies to that to date, but when you go back to the slide I showed that I said was complicated, where it showed the epidermis and the dermis, and you saw the IL-17, the IL-4, the IL-13, and atopic dermatitis. I mean, we do have one biologic out now that blocks IL-4 and IL-13 that is doing great, having great results with patients with atopic dermatitis. So yes, we do have that. And I think there are more that are even coming out that will help with not only atopic dermatitis, but other allergic and immune diseases as well.
Okay, another question. Can having a dog help create a healthier environment for a child?
Well, that's a complicated question, Sally, because if they're allergic to the dog, then that's going to be a problem. But dogs do carry some good bacteria with them as well. So I'm going to go to my default answer, which is it depends. Well, no one likes the answer, but it's the truth.
Well, I think everyone always appreciates truth and candor. So there you go. Someone's asking about, can probiotics help with food allergies?
You know, there have been some studies that have looked at that, and they haven't had any true good statistical significance that I have known, that I have seen in my review of the literature on that. But I don't think that probiotics are ever a bad thing. Again, if you're not living that really healthy lifestyle with that really healthy diet, low stress, you know, yoga, meditation, etc., they're never going to hurt you. That's my best advice on that.
Great. Someone's asking, which genus of probiotic do you recommend?
So, you know, I like all of them. I think the lactobacillus is very important. The one strain I do not like is I don't like to have any strep strains in my probiotics. And the reason for that is because of other diseases that can be turned on if you're exposed to strep. I won't get into that because it's not pertinent to this conversation. I like the Bacteroides, I like the lactobacillus, and I like more than 250,000 colony forming units. So there's some over the counter that are very weak, and you want to make sure that you're getting one that's strong. And I'm happy to email Sally if they want to email you, and I'm happy to give you brands that I like, names that I like. I prefer to do that via email just because I'm not here to promote a particular company.
And we appreciate that because this is unbranded education.
That is correct.
I think this will have to be our last question is, please comment on the positive attributes of eating fermented foods like sauerkraut.
Well, as I showed you in some of the slides that I showed you, you can see that it does help promote the health of the microbiome, which again helps keep the immune system balanced, and that homeostasis between our immune system is trying to go to the benefit versus things that are coming from the outside world that are trying to cause detriment. Fermented foods have been shown to help with that. So I think overall the diet is something we all know we need to do better at, not only for obesity, but now what we've shown today is that the diet can help with the microbiota, which helps prevent diseases such as allergy, asthma, immune diseases, and cardiovascular, kidney, and liver disease. So really, you are what you eat. It's simple. We've made it complicated all these years.
Well, let's work on uncomplicating it. Thank you so much, Dr. O'Connor, for joining us today. If you could just advance the slide. At this time, I'd like to encourage people to download the handouts from your control panel. The certificate is only available on the day when you listen to the live presentation. If you have any trouble downloading your certificate, please email me using the link in your emails that you'll receive following this presentation. Please join us next month for our May webinar when Dr. Gary Stephen will present on COPD and the best care. That webinar will air on Thursday, May 23rd at 4 p.m. Eastern. You can register for this webinar on our website at allergyasthmanetwork.org. Look for education in the horizontal navigation bar near the top of the page and scroll to webinars. You can also view our archived webinars on this page of our website. Also visit our website for quality guidelines-based resources on allergy and asthma. If you can move one more slide for me, please. This will be the last one. Also access important medical information on allergies and asthma from our partners, the American College of Allergy, Asthma, and Immunology at allergyandasthmarelief.org. I'd like to thank Dr. O'Connor one more time for fabulous information and webinar, and I'd like to thank our listeners for joining us. And we look forward to having you register to be with us next time on Advances in Allergy and Asthma. This is Sally Schessler for the staff at Allergy and Asthma Network. We hope you have a great and a healthy day as we work to breathe better together.